Okay, so for this video, we are going to learn about Lac Operon. So Lac Operon has been proposed by Jacob and Monod in 1961. So this is to explain the regulation of gene expression in prokaryotes, which is bacteria. And they received a Nobel Prize for this. So what is operon? Operon is genetic unit that consists of two or more structural genes that codes for polypeptides and promoter genes that control transcriptional activities. So operon, also known as functional unit of genomic DNA containing a cluster of genes under the control of a single promoter. So we only have one promoter, but it controls many genes together. So this means operon. So all these genes, so for example, we have 2 or 3 genes, it will be transcribed together into one mRNA and then translated together into enzyme in the cytoplasm of bacteria. So it is different from eukaryotic cell. For eukaryotic cell, one gene only transcribed and translated to one polypeptide. But for prokaryotic cell, many genes, especially 2 or 3 genes, they transcribe together into one mRNA and translated into different enzymes. So bacteria can adapt to the changes in their surrounding by using regulatory protein to turn a group of genes in activated mode or inactivated mode in response to various environmental signals. So when the genes are transcribed, it only produces one single mRNA for all the genes in that operon. Okay, so this is the structure of an operon. It consists of a regulatory gene, a promoter, operator, and structural gene. So this is the structure for lactose operon, which consists of 3 structural genes, LAC-Z, LAC-Y, and LAC-A. Okay, so it can be found in the bacteria E. coli, Escherichia coli, in response to the presence of the sugar lactose. That's the reason why you call it Lac Operon. So the bacteria can absorb lactose and break it down for energy or use it as a source of organic carbon for synthesizing other compounds. So it begins with the hydrolysis of the disaccharide lactose into the 2 components of monosaccharide, glucose and galactose. So it will be broken down. So the enzyme that used to catalyze the reaction is called beta-galactosidase. Okay. So the operon that regulate lactose metabolism is E. coli. Okay. So the gene coding for enzyme for a particular pathway are clustered together and transcribed into one single polycistronic mRNA. So polycistronic means it carries information for more than one type of protein. So for example, 3 structural genes, LAC-Z, LAC-Y, LAC-A, 3 structural genes transcribe together into one polycistronic mRNA. But this one polycistronic mRNA will be translated into 3 different enzymes. So LAC-Z into beta-galactosidase, LAC-Y into permease, LAC-A into trans-acetylase. So the component and function of the lactose operon. Promoter is where our RNA polymerase attach and transcribe the gene. Operator is where the repressor protein binds. When the repressor protein binds, it prevents RNA polymerase from attaching to the promoter. So it is a region between promoter and the gene. Okay, so we have 3 structural genes which code for enzymes for metabolism of the bacteria. We have Lac Z, Lac Y, Lac A. Okay. So if E. coli is denied glucose and given lactose, it makes the 3 enzymes to metabolize the lactose. So we have 3 structural genes. Lac Z code for beta-galactosidase. Instead, we use the word encode. Okay, Lac Y encode for permease. Lac A encode for trans-acetylase. Function of beta-galactosidase is to hydrolyze lactose into glucose and galactose. So it breaks down the lactose. Function of permease is to transport lactose into the cell. Function of trans-acetylase is to transfer acetyl group from acetyl-CoA to beta-galactosidase. Okay, wait. Okay, so these are the structural gene, Lac Z, that encode for this enzyme and the function. Lac Y encode for permease, function. Lac A encode for trans-acetylase, the function here. Okay, so now the permease, it allows lactose to enter the cell. Okay, so regulatory gene is called Lac I. It encodes for repressor protein that binds to the operator and inhibit the transcription. So Lac Operon is said to be inducible. Inducible means it will be affected. It will be induced, okay, with the presence and absence of the lactose. So Lac I is the gene encode for repressor, and it will be transcribed into mRNA and translated into repressor protein. It is then bind to the operator and there is no transcription occurs. Okay, so this will be the operator. This is the repressor protein which will be bind to the operator later. Okay, so we now look at the first condition where lactose is absent. So when there is no lactose, repressor protein is bind to the operator. It prevents RNA polymerase bind to promoter. So there is no transcription. When there is no transcription, no mRNA produced, then there is no translation. When there is no translation, no enzyme will be produced. Okay, so the diagram here showing that regulatory gene transcribe. Okay, transcription, because this is DNA, right? Okay, DNA transcribed into mRNA. So the mRNA is single-stranded and it will be translated into the repressor protein. So this repressor protein can bind to the operator here. So when it binds to operator, it prevents RNA polymerase from binding to the promoter. So structural gene cannot be transcribed. So when cannot be transcribed, no mRNA produced, so no enzyme produced. Okay, so when the lactose is present, so this allolactose will be formed. Okay, allolactose is isomer of lactose. This isomer, allolactose, will bind to allosteric site on the repressor protein, causing a conformation change, which is the shape change on the repressor protein. So now repressor protein can no longer bind to operator, while RNA polymerase can bind to promoter. And it will transcribe the structural gene, Lac Z, Lac Y, and Lac A, produce mRNA in polycistronic form, and translation occur on the polycistronic mRNA to produce beta-galactosidase, permease, and transacetylase. Okay, so now when the repressor protein bind to the allolactose, this allolactose change the shape of the repressor protein. So now this repressor protein can no longer bind to operator because the shape is no longer fixed. So now RNA polymerase can bind to the promoter and it will transcribe the gene ahead, okay, to produce polycistronic mRNA, one single strand of mRNA, and translated into 3 different enzymes to perform the respective functions. Okay, so as a conclusion, when lactose is absent, there is no transcription, no translation occur. Okay, when the lactose is present, it will convert to allolactose. Allolactose bind to repressor protein, change the shape of repressor protein, prevent and it will not bind to the operator. So this RNA polymerase can bind to promoter and transcribe the genes into mRNA. So what is the importance of Lac Operon in our life? So the baby, they cannot digest dairy product, okay, cow's milk properly because of the lack of bacteria in their body. Okay, these bacteria, they have the Lac Operon. Therefore, baby cannot hydrolyze lactose. So you call these babies lactose intolerant. Okay, but European, they are more lactose tolerant. Okay, it is born to in their gene, while Asians are prone to be lactose intolerant due to high consumption of dairy in European diet compared to Asian diet. Okay, so in next chapter, chapter 8, we have more opportunity to understand Lac Operon, okay, in recombinant DNA technology. Lac Operon will be used to control the production of certain desired gene. Okay. Okay, so as a summary here. So remember here, the DNA. Okay, these operon things are DNA. So in our assignment later, when there is mutation on the DNA occurs, okay, you have to think of it. So if mutation occurs on the DNA, it changes the genetic sequence. So for example, we have GATC, So when mutation occurs means any of the nucleotides here are changed. So when the nucleotide change, mRNA produced also will be changed. So when mRNA produced change, the protein produced here also will be changed. So when the protein here change, are they still able to perform their respective function? Okay, so if it is not able to perform the function, what happened to the lactose? So you have to think of this. Okay, so mutation means changing on the genetic sequence on the DNA. mRNA is still able to produce, but it is different, it is changing. So what happened to the protein here? Okay, so that's all for this video. Thank you.